Thank you very much. Thanks for the kind introduction and the invitation to be here today. So I've been asked to go quickly through traditional genetic techniques, and quite difficult to do in half an hour, so I will focus mainly on prenatal diagnosis. So this is the agenda. I will go quickly through the cytogenetics with just a little bit of history, how indication changed in prenatal, so the introduction of molecular tests for rapid abnormality screening, and then I will go to how we went from chromosomes to microarray with pros and cons. So chromosomal abnormalities are common, about 1% of live birth, 2% of pregnancies in women above 35 years. They are also present in about 60% of spontaneous abortion in the first trimester, and most cancers also acquire structural or numerical chromosomal abnormalities. So prenatal diagnosis of chromosomal abnormality started in the year 60 when Valenti diagnosed the first trisomy 21 fetus in utero by means of amniocentesis. This was the 1969. Since then, karyotype analysis has become the routine standard in prenatal diagnosis in the second trimester with amniotic fluids sampling until then. Brambati and Simoni were the first in 1983 to demonstrate that this could also be done in chorion biopsies and in the placenta, and bringing this into the first trimester. So these two assays has been and still are the standard of care for prenatal diagnosis of chromosomal abnormalities, together with cytogenetic analysis of the samples that are obtained with these techniques. So cytogenetics needs culture in fetal cells in prenatal diagnosis, needs culture in fetal cells for about 10 days, maybe a couple of weeks, so that then we can fix these chromosomes in metaphase, stain them with different technology to allow banding, and under light microscopy you can then analyze number and structure of the chromosomes. Over the years now we well know the accuracy of karyotype analysis is extremely high, a little bit less for CVSs, and this is for mosaicism, which you will hear more about tomorrow from Francesca. Main drawbacks here is the cytogenetic analysis needs trained people, and the training is also very long and time-consuming. The long culture, same culture, and this applies to prenatal diagnosis, leads to parental anxiety, and what we are definitely not really happy about karyotype analysis nowadays is the low resolution, which is limited to the resolution of the optical microscopy. So what changed? While karyotype analysis and cytogenetic has been almost the same for over 40 years, what changed in prenatal very quickly had been the indication to obtain samples, and especially with the advent of high-resolution ultrasound and improving on screening in the first trimester, second and first trimester then. And at the same time, we also know that these 5 chromosomes here, 13, 18, 21, X and Y, are by far the most frequent chromosomal abnormalities in prenatal diagnosis, which, depending on indication, are responsible of 95% or even more that what can go wrong in a prenatal sample. So they are still stuck cytogenesis with these 2 weeks of culture, which might delay therapeutic intervention. So this prompted in the 90s first, and then in the later on the use of alternative molecular methods for rapid prenatal diagnosis, improving prenatal management. So the first one has been the use of fluorescence in situ hybridization, These basically utilize bits of chromosomes which are labeled with different fluorescent dyes, so you can identify chromosomal specific region, and this is very good because also in a way improves the resolution of cytogenetic analysis, because you can detect a green spot or a red spot corresponding to a small deletion like the common deletion for the George syndrome, which would be invisible otherwise under the microscope. And the good thing for prenatal is also that probes works very well in interface nuclei, so you can just use the direct samples fixed instead of waiting for a chromosome to be visible in a couple of weeks. So this can be done in 1 or 2 days. Main drawback with FISH, especially in prenatal diagnosis, is again, one needs skilled operators. Also, interpretation of these two spots on an interface nucleus can be quite subjective. So it's labor intensive. It's better if is confirmed by 2 independent operators. Sample amount might be limited depending on gestational age and kind of samples that you can get in prenatal. Of course, the main drawback here is that it needs a specific indication. So you need to know what are you looking for and use the corresponding probe. So we did some quite a lot of work in rapid prenatal diagnosis, introducing the QF-PCR assay, which is a DNA-based test. So it's a molecular test, not molecular cytogenetics, exploiting genomic repeated sequences, which are known as small or short tandem repeats, STRs. These are 3 or penta or tetranucleotide repeated a different number of times. So the most individual will have 2 alleles, one of maternal, one of paternal origin, or different length. So the good thing is, well, these are quite easy to amplify by PCR with flanking primers, and if we label one of the primers with a fluorescent dye, then we can run these PCR products on a sequencer, and so we look at chromosomes in this way. Like a normal individual will have the 2 alleles of different sides and the same amount of fluorescence. Trisomic can be detected as 3 different peaks if the 3 chromosomes have 3 different alleles, or being quantitative PCR, if 2 alleles are the exact same size, they will produce a PCR amount which is double than the other one, 2 to 1, so with a clear different factor from the normal one. And informative, now they are not an issue anymore because we use so many markers that if one is homozygous and informative, we will have several more, other more on the chromosomes. And actually, most laboratories use several markers on the chromosomes. This is the one that we use on the X and Y, and these are the ones that we use on the autosomes. So just to show that, contrary of FISH, with this we can analyze the marker region all along the chromosome. So over the years we have seen that this also improves the detection rate of rare things like partial trisomies due to unbalanced translocation, for instance. This is how we detect trisomy 21. So the STRs can be multiplexed, analyzed in a single run. This is a trisomy 21 fetus with all the marker on chromosome 21 having abnormal patterns, while all the marker like chromosome 18 or chromosome 13 and the X chromosomes here are normals. So there are several advantages in this. Multiple losses analyzed, allows detecting partial imbalance. This is DNA-based test with microsatellites, so allows also maternal contamination detection. Actually, in Europe, this is the ideal test to be run before any other prenatal molecular diagnostic test, even a microarray, because you have to exclude the presence of maternal contamination in any prenatal sample. And using QF-PCR, you also, of course, detect all common trisomies. It's also been shown very useful for mosaicism. Molecular tests, you can include single mutation in the same PCR if you want. rapid objective and Situ automator, which allowed reduced cost and high throughput, being these now the standard of care in most laboratories in Europe, and I have to say almost everywhere in the world apart from the United States since 2001. From chromosome to FISH, but then we happen to have the full human genome sequenced and access to a genomic reference sequence. So these prompted developing a high-resolution molecular test using probes dispersed throughout the genome and fixed on a glass support, and these are the arrays. So we are through a step now going into the molecular cytogenetics. Advantages is well, the molecular karyotype identify both microscopic and submicroscopic imbalance. It is similar in principle as doing simultaneous FISH experiments with thousands or even millions of probes, depending on the array. And so the main advantage, especially in prenatal diagnosis, is that you don't need a specific phenotype indication to use the study, because you are going to analyze the whole set anyway. And also the analysis, contrary from cytogenetic analysis, there are now commercially available software which make things very, very easy for the laboratory. Results are totally objective, gains and losses are easily identified, and they always, of course, involve probes which are on the array, so known genomic location. And you can choose, as you will see in a moment, a different platform for resolution, density, and the design of this array. Drawbacks is, well, this doesn't identify balanced arrangements, which I have to say probably in prenatal diagnosis is more than an advantage rather than a drawback. May uncover unwanted information, so like it depends on the chip you use. You might see absence of heterozygosity, which may indicate consanguinity, for instance, so adult onset condition in a prenatal samples. Or identify variants of unclear significance, so things that can complicate the genetic counseling post-test, especially post-test with the expectant woman. So after the first proof of principle of using the microarrays in prenatal diagnosis, this was 2005, well, what we know very well nowadays is the cytogenetic analysis in postnatal diagnosis. Well, detects chromosomal abnormalities in about 3% of patients with intellectual disability and or dysmorphic features. While using microarray, this has gone up to about 15% of cases, which prompted the use of chromosomal microarray as the very first test in these cases already since 2009. In prenatal samples, well, we know now that about 6% more chromosomal abnormalities can be detected in fetus with a normal ultrasound and completely normal karyotype, and also about 1.7% in fetus without even karyotypes, so with just in low-risk pregnancy. So this prompted the use of prenatal diagnosis as a first-line test, microarray as first-line test, also in prenatal diagnosis in 2013. How it works is, in principle, is relatively simple. So you extract the DNA from the sample that you want to test and from a reference sample of the same sex. You label both DNAs with different dyes. In this example, one is red, one is green, and one is red. And then you put them, hybridize them together on a chip, which is designed with a particular sets of probes dispersed throughout the genome. So these DNA are going to compete for the same probes. and hybridize together there. This means that if you have an excess of patient DNA, you will have an excess of red. If you have, like is in this example, is a trisomy there. If you have a lack of patient DNA, you will have an excess of red, which is the control DNA, like in this example here is a monosomy X. So if you then scan the chip with a high resolution scanner, this means just take a high resolution picture and then computer software on each individual spot, which is the probes which are on the chip, can measure the amount of red and green and give a plot like this. So this is a chromosomal microarray analyzing probes on the whole genome, DNA starting from an uncultured amniotic fluid. And you can see very easily, you can detect things like this small deletion here, or in this case is a deletion in the duplication from an unbalanced translocation. And because you are analyzed the whole genome, you can also use this to identify something that you can find, like this was a very rare case of ring chromosome, mosaic ring chromosome identified by microarray and has been shown to be ISO 12P, so cause of polycythelian, and you can see how you can also detect these mosaics or those low-level mosaicism with the microarrays. So we are going from the common picture of cytogenetics of the traditional karyotype to the molecular karyotype, where you can go in depth, deep to see very, very small things, and how small depends on the array that you use. There are other array platforms which are less frequently to use, at least in prenatal diagnosis, and these are SNP array. There are basically two principles to analyze SNP across the whole genome. One is has been developed by Affymetrix, so this is just using several probes around the same SNP. So that each allele hybridize with a higher or lower efficiency depending on the position of the particular allele at the SNP locus. And then there is the SNP array from Illumina, which in principle is even easier, where the probes are stuck in beads, and then there is this is designed around the SNP, and then depending on the allele, only a single base is extended on the probe, and so then you only have a specific extension of that particular genotype. Either platform will give you a results similar data, and the main difference is that apart from a copy number, the same as you can have with a conventional array CGH, you also have genotype at the SNP site. This is an array with an isodisomy for chromosome 7, and you can see you only have the homozygosity for one of the two alleles, and there's no heterozygosity at all in the middle. So the main difference between the two platforms is, well, both are suitable, of course, for copy number gain and losses. With array CGH, you not always can detect triploidy, almost never. In prenatal diagnosis, this is not an issue at all because, as I mentioned, all samples have microsatellite analysis first anyway. Mosaicism are probably a little bit more easier to visualize using a SNP array rather than an array CGH, but probably array CGH has probably a higher sensitivity in detecting low-level mosaicism. And SNP array can also detect uniparental disomies, but only the isodisomies, not the heterodisomies. And absence of heterozygosity indicating consanguinity. This is not an issue, of course, if you use array CGH and not the SNP arrays. So as I mentioned, there are several different options nowadays for array studies. Basically, arrays can be classified for the probes which are used. Back arrays were much more frequent in the past. Now most, if not all, I would say all, platform employ oligos array, and then there are several different densities. So you will hear about array about 60 or array about 100 Ks. This depends on the number of probes that are on the array, and it gives more or different resolution. And then the distribution. So you can have probes which are equally sparse throughout the genome, arrays which have probes only in chromosome-specific regions, and array which are mixed using both things. So a backbone, what we called, and then a higher density in a specific region. And actually these are the most commonly used in diagnostic labs like ours, which we almost only use the 8x6 with an ISCA design. So it's a targeted array plus backbone for diagnostic region. And it's also interesting that not always the more is the better with the arrays, because depending on the array design, for instance, you can have, these are two cases, one with a small deletion, one with a small duplication, analyzed with both the 8x60 and the 180K and the 60, and you can see that in both cases you have actually more probes. So more resolution with the 60 rather than the 180. And this is because this is a targeted array, so with more density in specific regions. So back to ultrasound, but it's also interesting to see that independently from the array used, in prenatal diagnosis, mentioned that in abnormal ultrasound, it has repeatedly been shown that you can detect chromosomal abnormalities in about 6% more cases compared with cytogenetic analysis. And this is a back array, so targeted with a backbone, 6.1%, mixed array between oligos and back array at the beginning, almost the same extra detection rate. This is a SNP array, and in the NHD study of 2012. So different platform, but almost the same detection rate. The main difference is that depending on the platform, you can greatly reduce the number of doubtful, so unclear results, which in prenatal diagnosis is very important. And this brings to the last things about the array, which is the interpretation of the results. So basically, you have three kinds of reports. With abnormal results, this means copy number variant are identified, and these are clearly related to a particular phenotype, which is well described in the literature. Alteration contains dosage-sensitive or disease-causing genes, or if this occurs, the CMV is into a region of the genome which is very well known to be involved in chromosomal syndromes, or statistically more frequent in the patient populations rather than in controls. Benign results, well, this means that the CMV is not thought to cause an abnormal phenotype. The alteration is found more frequently in control populations. Well, there are more frequent polymorphism like this in different populations. And then the last, which is the main issue in prenatal diagnosis, is results of uncertain significance. This means the copy number variant has been observed is not related with a normal phenotype, but is large enough to be of concern. The alteration has not been reported in the medical literature or not found in available databases, does not contain disease-causing genes, and in this case, well, additional testing used to be required, evaluation of the family and also the family history to try to find out. these results, compared to when we started with the microarray, we also have to say that the more time pass and the more these variants are classified, they're either benign or pathogenic, or likely benign or likely pathogenic. So the number of these are decreasing, which together with the specifically designed or targeted array for prenatal diagnosis are trying to limit in these results to very, very rare event in a prenatal sample. And some such findings use also to be revisited over time as a new study are published. And also in postnatal cases, it's quite frequent to reevaluate patients that have been tested with fresh knowledge of the same findings. So this is how the testing strategy for for prenatal cytogenetics and for prenatal diagnosis of chromosomal abnormalities, including molecular cytogenetics. So in case of invasive procedure, we always set up cell culture as a backup DNA source, so if chromosome analysis is requested. In all cases, it is always useful to perform QF-PCR, and which can report abnormalities in 24 hours, so next day from the invasive procedure. This is a necessary step before any molecular test, microarray or single gene disorder in prenatal diagnosis, as maternal contamination can be detected. And if this is the case, in prenatal samples, you have still the option to go through the cell culture, which also used to select for fetal cells rather than maternal cells, so you can then retest the same sample after the culture, make sure there's no contamination there, and then go on with the molecular test. In this example is a prenatal microarray, which can be reported usually maximum a week after the fetal sampling. And then, well, if karyotype analysis is requested, the cell culture can be harvested to prepare chromosome, and chromosome can be also analyzed. With this, I'm finished. Thank you.